Beth Stroupe, and I am an associate professor in the Department of Biological Science, as well as the Institute of Molecular Biophysics. Like most tenure track and tenured professors, we're responsible for teaching, research, and service. So I have aspects of all of those obligations in my job. My research, I have a group of 5 graduate students and 3 undergraduates. And the primary focus of the lab is to use structural biology techniques to understand the protein molecules that are involved in sulfur metabolism. So I can break that down a little bit. All of life, from viruses to bacteria to multicellular organisms like humans, are made up of large chemicals that we call biomolecules. And those biomolecules have shape to them, and the shape that they have determines what their role is in the cell, what their function is. And so there are methods that we use to understand the relationship between the structure and the function for these biological molecules. The techniques that we use are just basic biochemistry, so analyzing the activity of the enzymes, to X-ray crystallography, which gives us a real zoom in to see the atomic organization of these very complex, very large molecules, again, that are the do all of the things that a biological organism needs to have happen. So we use X-ray crystallography as well as cryogenic 3-dimensional electron microscopy. So we use a combination of those techniques. And again, the topic that we're interested in are protein enzymes, so protein catalysts, that are responsible for converting sulfur into the form that can be used by organisms to build their biomass so that they can grow and replicate and function. My favorite part is analyzing the structures that we determine. So once we've done all the hard work and we have a finished product, we can actually visualize, like I said, at the atomic level, what the chemical structure of these very complicated chemicals are that make up all of life, right? So we're not talking just a couple of carbon atoms here. We're talking, thousands of carbon atoms, nitrogen atoms, oxygen atoms, water molecules that are arranged in 3-dimensional space. And so looking at those and seeing what the chemical relationships are that make these protein catalysts, these enzymes, function the way that they do, that's super fun. It's also super fun when my students start to do that and I can see them not just going through the steps of doing an experiment, but actually clicking and having new ideas and being able to put the pieces together on their own. So in the sciences, in STEM fields, typically there is a one-on-one relationship between a graduate student and their major professor. And so some of the students are just finishing up their first year of graduate school. And in that first year, they did do rotations where they spent 7 or 8 weeks in 3 different labs. And part of that was rotating with me. And we decided that we had a good working relationship and they were interested in the questions that we were asking in the lab. And so they decided to join my research group. I have another student who's been with me for 3 years now. So he's finishing up his 3rd year and will enter his 4th year. Typically, graduate students are in a lab for 5 years while they work on their doctoral project, again, with this one-on-one relationship with their major professor. When I teach, I either teach the first section of Introduction to Biology, so it covers the molecular aspects, sort of biochemistry, cell biology, and sort of basic biological principles that are common throughout all of life. I also teach an upper division molecular biology course that focuses on how genomic information is turned into biological processes in cells. The upper division course is an elective. It in particular is not required for the students to take, but it's related to what I do in my lab, to my research. And so it's fun to teach things that are a little more specific and a little more detail oriented. So I grew up outside of Chicago. So I went to a public high school. From there, I went to Wake Forest University, which is a private school in Winston-Salem, North Carolina. Straight out of my undergraduate, where I had a major in chemistry and a minor in music. I went to the Scripps Research Institute, which at the time there was only one campus, and it was in, and still is in, San Diego, California. So I spent 6 years living in San Diego and going to school. And when I left, or when I finished up my research in Elizabeth Getzoff's lab there, I moved to Brandeis University and worked in Nicholas Grigoriev's lab there. I learned X-ray crystallography as a graduate student, and then cryogenic electron microscopy as a postdoc. So we used both of those techniques in my lab. Straight out of my postdoc, I came to FSU. So this has been the only faculty appointment I've had. And I've been here since 2009. So I come from a scientific family. My mother was a math teacher and my father was an analytical biochemist. So we've been thinking about chemistry pretty much since I was a kid, because Dad would come home and tell us about his work at the kitchen table. My older brother is also a structural biologist. So Mom won't let us sit next to each other at Thanksgiving because we just talk about work all the time. And that's not very interesting to anyone but us. So I always sort of had it in my mind that this is what I would do. The sciences at Florida State are, the departments are organized so that they have core facilities that have instrumentation that we need to do our research. So in order for me to do the types of studies that I do, I need to have access to an X-ray generator so that we can collect diffraction patterns from the crystals for X-ray crystallography. And I need access to a cryogenic electron microscope. And these are not small pieces of equipment. These are multi-million-dollar pieces of equipment that really require a very sophisticated and dedicated infrastructure. So back when I was finishing up my postdoc, FSU acquired a Titan Krios. FSU was one of the first universities to have this particular instrument, and it is the state-of-the-art instrument to do cryo-EM studies. And so when they had a job opening that actually had the instrumentation that I needed, and not just the EM and not just the X-ray, but both of them together, with the strength that Florida State has in structural biology, it was really a no-brainer. So I confess that the research is what brought me here. It was difficult to close down my lab, and it's been difficult to direct my graduate students so that they can continue to progress when really right now they need to physically be in lab and they need to be interacting with one another and they need to be doing experiments. So unfortunately, I'm not sure I have a positive answer to that. But I'm glad that they're doing the reading. They're working on their writing. Some of them are not native English speakers, and so this gives them an opportunity to really focus on writing and not just have it be a secondary thing that they try to cram in at the last minute. But yeah, it's going to be a long recovery. And we're small operation. The bigger labs are going to be able to open up and get off the ground a lot faster than we are when we finally do get back in the lab. So I confess I have some anxieties about it. And so we're trying to stay as a cohesive group, even though we're doing it over Zoom remotely. But yeah, it's going to be tough. I confess that I'm actually not in the classroom this semester, so I haven't been directly impacted by it. My focus this semester has been on my graduate students' education, so we're doing graduate tutorials with them. Although looking forward to fall, I am definitely going to be prepared to have my lectures pre-recorded on the chance that we're still stuck at home. I would say that it's the other faculty members. I really like the people in my department. I like working with them. Even when we disagree about things, we're always working towards what's best for the university and what's best for the students here. So I'd start with my colleagues, and I would follow that up with the students. The very first person who joined my lab was a freshman undergraduate. He was the first author on the first paper that came out from my lab. And ever since then, I've just really enjoyed the people at Florida State. It's a big university. There are a lot of students, but the diversity, I think, brings with it sort of a vibrancy, I guess, that makes it a really nice place to be. But in addition to FSU, Tallahassee is a really great place to be. You can get to nature 15 minutes away from the city, and you're out in the woods, and you would never know that you were so close to the capital of Florida.